So it gives me great pleasure, before we take the group photograph, I'm keeping you excited for that, or warm, whichever way we look at it. So our final presentation today is from Kate Button. And Kate, as you will soon find out, has also been very instrumental in trying to change how we do our science, but particularly coming up with concrete solutions on how we can achieve this, particularly dealing with undergraduates and working in collaborative methods. So our final talk, Kate Button.
Okay, so I'm aware that I'm the last speaker in between us and the panel. But I'm going to start with a conceptualization of my kind of idea of the wider research problem. We've heard about this lots already today. But essentially, we have a publication bias where we have a preference for publishing certain types of results, so maybe positive results rather than null results. We have a publish or perish culture, which as an early career researcher still on probation before I get tenure, I'm very acutely aware of at the moment, where our careers are often determined by the number of publications that we have and the impact factor journals that they are in. And that might drive the desperate researcher who is keen to pay their mortgage and keep their job into adopting certain practices that may enhance the probability of finding significant results, but perhaps at the cost of the reliability of those results. And we've heard about some of these already: low statistical power, poor control for bias, and questionable research practices. And all of this creates a climate, or a kind of a toxic environment perhaps, where we get to the situation where people like Johnny and Edie's might have suggested that most of our published research findings may indeed be false. So in 2013, we looked at estimating the prevalence or the median statistical power in the field of neuroscience using some of these meta-analytic techniques that Richard was talking about. But we used a very generic search term, neuroscience, with the tag meta-analysis. And we just wanted to do something very simple just to see how much power do studies with the sample sizes that they're using have to find the best estimates we have of effect sizes from a corresponding meta-analysis. And we estimated median power to be about 21%. And we know that low power increases the risk of various types of inferential error. So it increases the probability of a type 2 error, but they're less likely to make it into the published literature perhaps. But it also increases the probability that a statistically significant finding is what we would call a true positive. And we also know that small studies yield imprecise effect estimates, so we tend to get more bias in the effects that we're estimating. And when we add the selection filter of publication into play, then it means that we tend to get estimates that are biased in a positive direction and more likely to make it into the literature. We've also heard previously about Simmons et al.'s famous paper, where if we adopt several analyses paths in an afternoon, and this could be a couple of hours' work, sat at your computer, we can get to a situation where very quickly we've got about a 60% chance of finding a false positive finding, perhaps. So we know that it's very easy to interrogate our data, and it's very easy to generate positive findings. Whether they're reliable or not is another matter. So how does this all relate to student projects? Well, we can kind of think about the student project world as being the problem magnified. Each year we have undergraduates in our respective departments running a series of research projects. We might have 100 of them, for example, and they're running these multiple projects that are often poorly resourced. So they have very limited time to run these research projects in. They have very limited money sometimes, and you might have limited access to populations of interest. Historically, the assessment criteria have often focused on individual contributions and creativity and novelty, although I'm pleased to say that I think this is changing. This leads for the potential for multiple studies, which are small, potentially underpowered, and have the potential for poor design. They may be more likely to be testing novel hypotheses rather than replication, and then there may also be the potential for undisclosed flexibility in the way that students are analysing these data, and lack of transparency perhaps in the way they might wish to report them. So I'd like to kind of use an example that we gave in our 2013 paper that I've adapted slightly for the the undergraduate dissertation setting. So I'd like you to just imagine that we have 90% of student projects are testing hypothesis where the null hypothesis is in fact true. We set the significant level to the 5% alpha that we usually use, and we have the average statistical power of these undergraduate projects set at 20%. If we do 100 undergraduate projects, and this might be kind of the norm for many university or psychology departments, then we would expect 100 true associations to exist, and of those, the studies would have, with 20% power, will have, on average, be able to detect about 2 of them because of the 20% power. Of the remaining 90 non-associations, we're going to falsely declare about 5% of them as significant, so about 4 to 5. Now I'd like you to imagine that those students that were lucky enough to find a statistically significant finding are encouraged to go on and write those up for publication. 2/3 of those will actually be false positive findings. So I think we might be sending the wrong message to our students very early on in their potential careers, where we're rewarding luckiness or the ability to find chance findings, perhaps over rigorous methods. Because now I'm sitting on the other side of the divide, and I'm a lecturer, and I'm interviewing potential postgraduate students. We know that undergraduate degree class, whether you have a master's and whether it's a distinction, but also whether you've managed to have an undergraduate publication is really key in the way that we determine who goes on to study within our universities. And we know that we're working in a competitive environment, and it's becoming increasingly so. So if we look at this figure here, we have along the bottom number of years, and the orange line is the number of faculty positions, cumulative year on year. And the blue line is the number of PhDs awarded, cumulative PhDs awarded. And we can see that over kind of recent years, there's been a massive increase in the number of PhDs that we're producing, but the faculty positions are remaining relatively constant. So it's getting harder and harder for us to get tenure or permanent positions within this sort of environment. And what predicts the probability of us making it as successful PIs or academics? Well, this was some really interesting work done in 2014 by Van Dijk and colleagues, where they looked at 20,000 authors on PubMed, and they looked at what predicted academic success. So the red lines are those that go on and stay in academia and become PIs, and the blue line are authors that will leave academia. And unsurprisingly, the 2 biggest predictors of whether you'll make it in academia are the number of first author publications and then the impact factor of the journals that those publications are published within. And if you're interested about issues about women in science, whether you're male also is pretty high up there too, but that's a whole other talk. So is that a problem? If we're rewarding people with high impact publications, is an impact factor a really good indicator of whether those are high quality studies or not? Well, probably not. I've got the figure at the bottom here is a nice bubble plot by Marcus Manafu, where we have impact factor of the journal along the bottom, and then we have the degree of bias in the effect estimate on the y-axis, and the size of the bubble denotes the size of the sample from which the estimate was drawn. And what we can see here is we have a nice correlation with impact factor and degree of bias, with some of these studies with very small sample sizes and a huge degree of overestimation of effect size getting rewarded by publication in some of the highest impact factor journals. So we can think about the wider reproducibility problem as a current system that rewards positive findings and novelty over rigorous methods and replication, and that a lack of scientific rigor in the way that we do things means that we are more likely to be able to find those positive results, but at the cost of them perhaps being more likely to be false positive. What we really need is to align training and incentives for career progression with scientific rigor, not results. So in 2013, we made some recommendations for researchers where we suggested that some ways to tackle this, and this was based on best practices from across other scientific disciplines, was performing a priori power calculations. You know, you can find that in any basic psychology textbook. This isn't new. This just perhaps we forgot to do it for a while. pre-registering study protocols and analysis plans. We've already heard about the Open Science Framework and registered reports, but this has been, this was based on work that had been going on in the world of clinical trials for some time. And then thinking about how we can work collaboratively to increase power and replicate findings, where we're working in areas where on our own we will be unable to get sufficient sample sizes to meaningfully answer the questions we're interested in. And this has really been pioneering, this has really been revolutionary in the field of human genetic epidemiology, where they're really making great strides now in terms of genetic consortia and so on, and it's completely revolutionized people's understanding of the genetic architecture. We more recently published our manifesto for reproducible science, which has already been mentioned, and in that we made proposals for the need to improve methodological training. And I suggest this can really start at the grassroots at the undergraduate level, because by training our undergraduates to do this, we're also going to be, de facto, training ourselves into these processes as well. We think about collaborative and team science and promoting study pre-registration, and all of this can help us improve the quality of our reporting. So, in 2016, we published a commentary in The Psychologist called Instilling Scientific Rigor in the Grassroots, Feasibility of a Novel Multi-Center Methodology for Undergraduate Psychology Projects. And the idea was that we could kind of embed or use some of these methods that we've been thinking about for use in our wider research into how we can improve the training of our undergraduate students. So we can improve the quality of research training in open science and hopefully improve the quality of the research outputs, both current, so in terms of the student dissertations and any potential publications, but also by training these students so when they go off, if they do decide to embark on a career in research, we've already embedded some of the key principles of open science in them, and they can take that forward with them and hopefully inform others of the process as well. So this was the structure of the first undergraduate consortium that we run. So this was a collaboration between the GW4 universities, and the 3 universities out of the GW4 that took part were myself at the University of Bath, Christopher Chambers, who we've already heard about today from the University of Cardiff, and Natalia Lawrence at the University of Exeter. And we each of us had a number of students, so there were 8 undergraduate students in total, and we decided to all work together on this one project, the effects of training response inhibition on self-reported liking of unhealthy foods. And it was a replication study, but it was also thinking and investigating moderators of a previously found training response. So I just added this slide in earlier today, since this talk is at the BPS, and I think it was a year and a half ago now, I spoke at the Undergraduate Education Committee meeting in I think it was in Birmingham, and at that point it was still in the guidelines, the accreditation guidelines, that it was very much focused on individual contribution to student empirical projects. But I'm delighted to say that in the revised 2017 version, we now have this statement at the bottom that students may undertake their empirical projects as part of a project or laboratory group, provided they are still required to demonstrate the above skills individually. So the whole thing I've been grappling with this initiative is thinking about how can we satisfy the need for individual assessment at the same time as promoting and training and delivering reproducible open science. And one of the ways of doing this, well, the way that we've approached it so far, and I'm pretty sure there'll be many others, and I'm looking forward to seeing how others take this idea forward and adapt it and improve it and so on. But really, the core idea is that it takes a lot longer to do reliable, reproducible research, because each stage requires more thinking, more careful planning, and so on. And actually trying to cram that all into an individual student project in their final year is very challenging. So what we did was we researchers spent from January to September the previous year thinking about a main research question and an overarching study protocol. So this was a bare bones protocol that we developed. We then gave that to the respective students over the summer and said to them, okay, take this protocol away and think about what additional hypotheses you would like to add to this protocol, and tell me which measures you would need to include, and design a data analysis plan to test your individual hypothesis. We then had the first consortium meeting in October, where the whole consortium met together, and the students individually presented their proposed hypothesis, the measure that they proposed to add into the kind of research proposal. And as a team, they collectively decided which of those to include in the final study. We then registered that protocol on the Open Science Framework, data collection commenced, and then the students were provided with the data at the point of when they needed to start writing up their dissertation. And because each of them had contributed their own idea to the project, it meant that we were able to meet the BPS requirements of being able to assess students within an individual site individually. So each of their dissertation takes a slightly different form to the other student because they've got a mini question within the overarching project, but they're still contributing to an overarching whole, as it were. And then in April, we have a consortium meeting for the results. So again, the students then prepared a mini presentation on their own study hypothesis, but then were also able to contribute to the discussion about the overarching conclusions for the main study. And that way, at each stage in the protocol write-up and the consortium meeting too, they're able to contribute meaningfully so that they can meet the criteria for authorship of any research outputs. So, trying to think about how we can align this need for individual assessment with trying to do rigorous science, the first approach we took was a replication and extension. So, Christopher and Natalia have previously published a study looking at the training response inhibition to food and whether that's associated with weight loss. And we thought about whether we could replicate that finding and then thinking about how we could extend it in terms of the students could think how that training effect could be moderated by other variables. So we performed a sample size calculation based on the treatment effect from that original paper. And we needed 106 individuals for that. And then each student contributed their own moderator hypotheses. So that's kind of secondary analyses. So that upped our sample size to 412, which you can tell that's a rookie error from a very new lecturer. It's very ambitious for an undergraduate project. So by the time they finished data collection, we'd actually only reached 238, which is large enough for the replication analysis, but was insufficient for the moderator analyses. So we've decided to continue data collection beyond the student projects, and we'll obviously adjust for the interim analyses we have now conducted as part of their dissertation. But we're going to continue collecting data until we reach our sample size of 412 before we write this up for publication. The students collectively wrote the protocol, and they each contributed their own a priori statistical analysis plan for their individual hypothesis within that study. And that's been published on the Open Science Framework, and the DOI is there. And because each student contributed to that, they qualify for authorship, and that's also something they can now add to their CVs as a CV point for when they go for future jobs and so on. And by having this pre-registration, students were able to follow the statistical plan that they set out in that protocol, which actually made their write-up easier. So they did a lot of thinking at the front, and it was quite hard work, and I think at the time they felt like they were at a bit of a disadvantage to their contemporaries because they were all going ahead collecting data, whereas our guys were still struggling to kind of get the protocol ready for registration on the OSF. But when it came to the end of the project, they'd already written their methods and their introduction in their methods, and they'd already drafted their results section because they already knew exactly how they expected their results section to look. So it was just a case of running the analyses, which isn't trivial, but running the analyses and then interpreting and drawing their conclusions. So it's actually quite an efficient way of doing it, but it doesn't always feel that way at the beginning when you're struggling to get your study set up. We're also intending to publish our final data sets on either a university repositories or on the Open Science Framework, and where possible making study materials available on the Open Science Framework as well. So to conclude, there are solutions to the problems of reproducibility, and I actually think I agree with Daryl. This is a very exciting time to be a researcher in psychology. I think we're making great strides, and there's lots of changes happening across the board, which are very exciting. But we have to be mindful that rigorous research does take more time and it does take more resources. So perhaps we need to think a bit creatively about how we can work together to pool resources to address some of these issues. But also systemic change is really happening. Funders, publishers, researchers, they're all moving in this direction. So really we need to ensure that we're preparing our students for the future of open science, because it is coming, if not already here. So consortium studies offer a way to tackle these issues. Students participate in and train in rigorous methods from the start, and the idea is that we can start instilling best practice in the grassroots to try and change scientific culture from the ground up. So I have some acknowledgements of these are the 3 consortium that we're running at the moment. Well, the 2 of them are in our second year of the GW4 one, and I know we've got questions right at the end, so I'll finish there. Thank you very much.